Hi, good morning, and welcome to the ZP Developer Zone webinar. So we do this webinar every Thursday at 8 a.m. London time, and it's really just answering sort of questions or that come in regarding ZP and ZP products for the week. So let me go rather quick. Last week I spoke very fast because we had a lot of meetings. This week I'm going to speak rather fast because we have a lot of questions. So we do have the ZP Academy if you're looking for free advice or free sort of courses. There are a couple of free courses on the ZP Academy about what ZP, the kind of analytical technologies that ZP uses. And I will, how do I put that? There's a couple of courses there. One for introduction to biosensors and one electrochemical techniques for biosensor developers. And we do have our webinar. This is an example of that webinar that we do this every Thursday at 8 a.m. London time. Do have collaborations. These collaborations are typically happening in the UK and in Norway with academic institutions because we can basically leverage that funding. We do make job offers. We have been recruiting in Norway of recent, and we do have the ZB Developer Zone website. So and of course, finally workshops. Now workshops, we've just updated this. So this year. Each one of these little sort of buttons, and if you see a link, by the way, the link is definitely underneath the. If you see a link, the link is definitely underneath this video on YouTube. But we've held a whole series of workshops so far this year, and these are really around sensors, IVDs, and even wearable sensors. So I think there's a lot of kind of scope. I think people really enjoy them. And we've got two in June, for example, one in Norway and one in the UK. And again, if you see a link, then I've already put the link underneath the video. Questions. So somebody's interested in sensors for Troponin I. Troponin I is a biomarker for sort of cardiovascular disease, cardiovascular events. I'll discuss that. Somebody else was interested in equipment for platinum group metal detection, PGM. We will talk about that. Somebody also wanted to know the operational life of a CGM, a continuous glucose meter. What you're going to find interesting this week is a lot of these questions. They're different questions, but they're there's a lot of interrelation. So later on, you'll see that somebody's very interested in sensors for blood gas analyzers. And so the question number 3 is very pertinent to that person as well. So question number 2 and question number 4 seem very different. Question number 2 is about what's the equipment that you use for platinum group metal identification and quantification. And question number 4 is the use of Cyclic Voltammetric Stripping for PCB manufacturing, printed circuit board manufacturing. You'll find that actually the technology is the same. It's only the kind of method files and the analysis files that have actually changed. But otherwise, technology is very similar. Technologies for Propofol. Several of us ZP actually have backgrounds, obviously, in electrochemistry and in biosensors. But fortuitously as well, many of us actually have. I have a strong background from the pharmaceutical industry and from graduate work in organic chemistry. So we do like the electrochemical detection of some of these. I would describe it really as a phenol, substituted phenol. So propofol is, I think I want to say it's a sort of, it's an anesthetic used in operations, and how do you detect it and make sure that the patient is within safe limitations. Somebody wants a custom screen printed electrode. We get a lot of this kind of questions, so I will address that. Somebody else just wanted to know the range that a fructose sensor works in, and I think I'll show you some data today that says sort of 0, 5, 10, 15, and 20 millimolar. So I generally hope that that will help answer that question, so I will do that. Sampling system for biosensors. This really plays into some of the environmental applications. How do you bring a sample to sensor? Functionalizing a screen printed electrodes. There's a team that we're supporting in Southeast Asia where they're trying to modify one of our hypervalue carbon electrodes. The nice thing is they've given us some really detailed slides, so I'll definitely discuss that. Measuring pH and oxygen on an Arduino. Sensing systems for total antioxidative status. TAS, I will discuss that this morning. And sensors for blood gas analyzers. So there's a lot of questions, so I suggest that if you can spot your question there, that you may have to sort of fast forward through the video to kind of find the relevant parts or the relevant question, let's say. question number 1 was really about troponin. So they want to know about our troponin sensor. Do they need reagents to run the sensor, and does it need calibration? ZP, at ZP, we're really steeped in really technologies from the glucose strip world. And what I mean by that is, I sort of, in my career, I've worked manufacturing glucose strips. And so it's just in our mindset that you don't need reagents and you don't need calibration. It's just the way our minds work. For me, calibration is really done in the factory. It's about manufacturing repeatability, and it's not something that you get the user to do in the field. When you're doing that, then you're really struggling. So no need for additional reagents and no need for additional calibration. I'll just slightly drag myself out of the way at the moment on this. So we have tested these sensors. Here, I think we did a test where we were sort of testing in triplicate of a troponin I at 0 picograms per mil, 1 picogram per mil, 10 pico 100, 1,000, 10,000, and 100,000. And we also tested in spiking human plasma as well. We just went sort of very low, which is 0, and very high, which was 100 in that case. And actually, I think we were able to distinguish 100. So these troponin I sensors are kind of a form of immunosensor. This really plays into, when I say plays into, it's related to somebody that's actually struggling later on in this webinar to make some immunotype sensors, and I'll discuss that. But anyway, this is an immunotype sensor, and where the troponin I is kind of specifically binding to its antibody in this case, and that's essentially how we generated the data. In terms of productization, we are able to obviously put these kind of sensors into microfluidics. We're really good at, again, I think I've mentioned glucose strips earlier on. We're really good at making microfluidics as simple as they need to be. I think people really misinterpret this kind of lab on a chip concept in microfluidics as literally I'm going to translate the lab onto a chip. It's like, no, honestly, you do not want to do that. You want to keep it much more simple than you would have otherwise done in a lab procedure. But anyway, we're able to place these sensors into microfluidics. And something that struck me as well is when I was putting together this is that we're really good at sort of multi-analyte panels and multi-analyte tests. We have essentially hardware. Again, if you see a link, then you'll already find that these links are underneath the video, and you'll be able to navigate to that particular page. But here you can see one of our scientists actually, they have 6 electrodes in front of them, and they're basically just testing them. So we're really good at multi-analyte testing. There's a couple of videos. One of them is just a quick demonstration. Let me just silence that. We tend to do this kind of simple microfluidics. This is a kind of credit card format. The reason I brought this up was a little bit was because sometimes we definitely have a philosophy at Zimmer and Peacock about MVP, minimally viable product. Really, just how quickly can we get somebody into a clinical setting? And sometimes you have to pull tricks. And I say tricks, things like you could, for example, run more than one sensor on the same sample and just sort of start to use averaging, etc., to try and kind of improve your accuracy or your precision. But we do have a lot of hardware all ready to go. And I think this is a really good one because we literally had somebody come to us about Christmas time, and last week they did their they did their demo for their investors, and they really passed with flying colors. And this is kind of what ZP is all set up to do. So for example, let's say somebody wanted to have a minimally viable product. They wanted to get into a clinical setting with with a troponin sensor. Then what ZP has is we have sensors obviously, and we have hardware. We have app development. We already have this platform where the app is actually able to talk to the cloud as well. Now, don't worry, when we're doing things like preclinical studies and there may be real patient data there, it doesn't demand the patient's name, the patient's age, etc., because now you're getting into kind of problems with you can't store patient ID data in the cloud, or you can, but you have to sort of be really careful about it. But we're really literally just using patient ID and sending the patient ID to the cloud. But the system that we have is you can have a sensor, for example, it could be troponin I, you could put the sample on there, it could be human serum, for example, The app will then tell the sensor, do this, and the sensor will essentially do it, and then the sensor sends the data to Julie. So it's really useful for people in preclinical kind of settings, because actually they get the data in real time up in the cloud. So even though we're able to give, let's say, the health practitioner, you would often blind the health practitioner because you wouldn't want them using these numbers unless you had some sort of clinical approval. So you've got to be careful of that. But we can give real numbers on these devices in real time, and it's just a strategic decision whether you do. But what I'm saying here is actually the raw data itself is actually in the cloud, which is fantastic, because it stops things like people are not using the sensor correctly in the field. You don't know about it for several weeks, and then you find out that all the data is rubbish, or you're just getting complaints. And so being able to see the raw data as it's collected in the hospital setting and it going straight to the cloud is a real boon and benefit. And that's just come from experience, that we've obviously gained. I think this is our 9th year now at Zimmer & Peacock. So you can work with Zimmer & Peacock, including our Troponin sensor, micro projects, mini projects, proof of concepts, proof of principles, and MVPs. I'll put these links here. Really, I think anyone who's doing a medical device, whether this is Propofol or whether this is Troponin I think the sort of MVP last link in the list here is really the kind of one to pursue. Question number 2 about platinum group metals. Could you give me information regarding the detection of platinum in solutions? So I've got a couple of videos here, and I definitely have links for them on the next slide. Let me just silence that. And so these couple of videos, and I definitely have links for them underneath. What you'll notice is that we're actually using the same workflow that I was just discussing a minute ago, where when I say workflow, I should say sort of hardware. We have a sensor. It's obviously not the Troponin I sensor this time, but it's actually a platinum group metal sensor. We have the meter, we have the apps, and we have our cloud. So we're able to kind of put a sample like 50 microliters, for example, if there's no microfluidic device on there, which is not in this case, just put a drop on there. The app on the phone tells the meter what to do, and the meter gives the data back to the phone, where it can actually display the concentration. Often in sort of metallurgy, it's often ppm, but we change the units depending on the application. So, for example, in agriculture, there's a unit called kilograms per hectare. In chili sensing, it can be SHU, Scoville heat units. But and this comes to the point earlier on. Actually, somebody asked us, whether we need to calibrate. No, we do all of our calibration in the factory, so that the sensors come out in a repeatable manner. But yes, the hardware that's involved is a sensor, a meter, an app, and a cloud. So in fact, all the data is stored up in the cloud anyway, which helps troubleshooting and quality control later on. And I should say as well that actually Julie, the cloud system, will actually give you a report as well, or a certificate of, when I say analysis, certificate that that you can sign this off, and that's the result. And a couple of useful links. And like I say, any links I will have already pasted them underneath the video. Question number 3 is quite simple, short question, so I'll answer it quite quickly. But this question number 3 is also relevant to people interested in using glucose sensors, for example, in blood gas analyzers. It's a simple question. How many hours can you measure glucose continuously? And so I'll answer it in a different unit, which is days. So we've been running a glucose sensor for over 125 days, and it's still operational. So many of you who've sort of listened to this, I say this a lot, but glucose sensors are very robust, really no issue. So blood gas analyzers, great, but days and days and days and months of operation on this sensor, we've never actually stopped running it. I've been showing this slide for a good 6 months at this point, and it carries on working. Question number 4 was really about Cyclic Voltammetric Stripping. So one, a big fabrication technique in this modern world is printed circuit boards, PCBs. Some of the processes to make a PCB are electrodeposition. And so we have a sensor for measuring the performance of, they're called plating baths. So let's say you wanted to put copper down on a printed circuit board. You want to understand how well your bath is functioning. So we have a technology which We have electrodes or sensors, let's say. We deposit the copper on those electrodes, and then we reoxidize or oxidize the copper. Yeah, reoxidize is the right term here. Back off the surface to actually tell us how much copper, or how well rather, the copper was actually depositing on the surface in the first place. The voltammetry part of this is there's a voltammetric excitation of that electrode, and we get a stripping or the removal of the metal, and the sort of area under the curve, or the peak height is peak position is indicative of the metal, and peak height and peak area is indicative of how much there actually was. And so it tells us not only what the copper concentration was, but really how the levelers and brighteners, the additives, and the suppressors that are also in the bath were performing. So it allows, sort of, I would say, sort of near line quality control, make sure that that bath's running correctly. Again, any links that pop up, I will have put them underneath the video already. This is similar to all of the conversations earlier on about, we have a sensor, we have electronics, we have an app. You can see it's just the app that's changing every time. So actually the sensors also change as well, but we have a platform, and this platform really helps people get to either products. We make products upon it, but it also lends itself into the startup community that needs to do minimally viable products as well. And you can see the fact that we reuse this technology so much gives you a sort of sense of robustness. That sensor is in meter, sample goes on to sensor, app is run, app tells meter what to do, app is able to give result, but app also sends data to the cloud where we can store and use it for quality purposes. This one is on Propofol. So at Zimmer Peacock, we do have a long history now, almost easily over 5 years of analyzing many phenol compounds. Propofol is an example of a phenol. This is a sort of anyone who's particularly interested, I think it's what they would call 1,3 substituted phenol, and 1,3 dialkyl substituted phenol. And these phenols, because we're pretty good at the functionalization of electrodes, including layers, and we're also very good at the excitation of electrodes, and also importantly as well, the interpretation of signals, that we're able to kind of work with molecules such as this. But what makes us different, see, it's not just sort of making a sensor and doing it in the lab. We're actually able to take these kind of technologies and again put them on the platform. I haven't really mentioned it, but the platform is called Sense It All. Take a sensor that's specific to the analyte of interest, put it into the meter, run the app, so the app tells the meter what to do. So immediately then the person using the app, as long as the calibration is done correctly, will get a result. As I say, calibration is not done in the field. Our manufacturing is repeatable enough that, in fact, we can manufacture in enough reproducibility that you can calibrate in the factory, and all the calibration factors are taken with you. I think this video is actually useful as well, because actually you can see that we know it's not acceptable in a final product, but in a minimally viable product, we actually have a QR code here, so you can understand that actually we can carry calibration factors out to the meter as well. So sample goes on there, human sample would go on there. App would tell meter what to do. Meter gives the data back to the app. The app again sends it to the cloud. This is really useful in small preclinical trials, maybe even clinical trials. Again, you don't put the name and age, etc. of the patient. You can just put a patient ID. We've been doing this kind of work quite a lot with the NHS, the National Health Service in the UK. So we know that workflow is acceptable, and we're able to give a result. And we're importantly able to put a result in the cloud. It is important because then your sizzle sitting in your office, and you're able to see that the clinicians, as the clinicians do the test, then actually the results are kind of popping up on your dashboard as well. Question number 6 was about screen printed electrodes. This is one of the toughest. We do get this question a lot, and I think that the problem with custom screen printed electrodes is, at Zimmer Peacock, we make screen printed electrodes, we do custom work for people, but it's a lot of effort has gone into our screen printed electrodes. So when I say, and I've said it a few times today, that we don't need to do calibration in the field. Actually, it's in part because the screen printed electrodes that we're manufacturing are so good. We have a sort of RSD, residual standard deviation, not relative standard deviation, or coefficient of variance of 5% or less. That really helps us not need to calibrate in the field. I think with some manufacturers, their underlying variability of electrode means that you have to calibrate in the field. Anyway, the problem with custom screen printed electrodes is when you make a custom piece, you're not able to invest the years of effort that we have in order to get repeatability. So I think the biggest problem with custom screen printed electrodes is just quality, and it's quality and reproducibility. But this person has a couple of likes. They'd like to use our hypervalue geometry, which is completely fine. But rather than having silver chloride, they would rather have carbon as the counter electrode. I haven't really got the time to go into this today, but we've chosen silver chloride as our counter electrode. Not because of some quaintness or something. It's really a good decision. So if people want to go from silver chloride to carbon, because basically it's probably just historical for some people. Or maybe it's an essential, but I suspect it's historical. I would stay with the silver chloride as the counter. Anyway, they want to use a titanium oxide paste as their working electrode. They'd like to do it on glass. And the reason I'm sort of not jumping over this is because I got a glass, you can print on it, but and I actually just realized as well that people can be quite good at printing. I've definitely know that people are printing silver tracks on glass. And it's something that just occurred to me is that you may have to heat it quite hot, and cure it at quite high temperature. Carbon ink just can't take it. So I'm really reluctant to sort of do it because I think it's going to be quite troublesome, and we'll spend problems with temperatures, problems with adhesion. My honest advice is fail fast but fail cheap. So I would honestly take one of our hypervalue electrodes. If you really want a carbon electrode on there, it sounds ridiculous, but we've got a paintable carbon ink. I'd actually literally paint it. I'm only talking about producing a few prototypes, but I think it's better to try out a few prototypes that are kind of handmade as opposed to going down the more expensive and probably tortuous route of customization. Then you hand paint on carbon electrode onto that counter electrode if you really want it, and then hand paint your titanium oxide paste onto the working electrode if you really want it. The other route is do a sort of mini project with Zimmer and Peacock, but I can't advise it. We've spent a long time getting reproducibility on our electrodes, and I think there's a sort of false story put out there that you can just go out and screen print the stuff. Yeah, you can. It may even look good, but it just won't function well, and it's probably the area of electrode that will be fairly irreproducible. Question number 7 is an easy one. So just really wanted to know the working range of your sensors. So I just pulled the data off the website, so I can see that we've tested at essentially 0, 5 millimolar, 10 millimolar, 15 millimolar, 20 millimolar. If that's not within the range that you're expecting, it's okay. If you're doing a sort of, let's say you were doing an analysis of fruit juice and the fruit juice was more concentrated than 20 millimolar, then you would design a workflow. You said take 1 volume of the fruit juice, mix it with 10 volumes of a buffer, and then test that. As long as you take into account in your calculations that you did a sort of 1 in 10 dilution, then you're completely fine. That's something that we do a lot of as well. But the raw sensor itself can run from 0 to 20 millimolar. This is just something that people ask us this a lot, is really kind of, especially in sort of environmental applications. So we made a little rig in order to bring samples to sensors. I'll sort of run through this. This is the reservoir. Let's say this is the reservoir. And in there's a sort of liquid sample, but we want to bring the liquid sample to the sensor. So we've got what's called pressure pumps and vacuum pumps. They're just sort of, obviously pressure pump by the name applies pressure to the reservoir, and vacuum pump applies vacuum to the system. And when you've got a couple of valves, you can use sort of valve logic in order to be able to suck up solution to the sensor or to send solution to waste. And this is the sensor itself. I think this is a point actually I should, when you've looked at, if you're watching this webinar today, you'll see that a lot of our electrodes often look the same, they're often the same electrode but functionalized at the very last minute to be something different. And it's this kind of breadth of application on these same electrodes that means that we're manufacturing quite high volume, and we get really good reproducibility and repeatability of manufacturing. And then that all plays into this whole story about why we don't need to calibrate in the field. I will talk about calibration in field because somebody's going to talk about blood gas analyzers in a minute, so I will bring that up. But this was a rig that we put together which had pumps, fluidics, piping, electronics, reservoir, and we were able to bring samples out of the reservoir up to the sensor and then send the sample on its way to waste. I mean, this consists of, the apps, and I've talked about apps at the moment. So we have a core architecture of an app, and we can just change the app skin to reflect the customer or the application. Obviously, you have electromechanical engineers, so there's no problems in putting together pumping systems, and we have our electronics. And lo and behold, I realized when you look at the electronics, you realize it's the same flipping electronics that he's been talking about in the last 8 questions. Question number 9, functionalizing hypervalue carbon electrodes. So at ZP, we do have a lot of academics and research groups that kind of come in, buy our screen printed electrodes. They're pretty good, and they want to functionalize them. And then I think what makes ZP a little bit different is that we are willing, once, as soon as somebody asks us a question, it's within the week then, because we're doing a webinar, we're able to kind of answer questions. But this is what's going on here. I advise them to, when Zimmer and Peacock is functionalizing something like a carbon electrode, we use a lot of cyclic voltammetry as process monitoring to make sure that we're actually functionalizing this. So they sent me a nice detailed list of what they were up to. So they took the hypervalue screen printed electrodes, they washed them, and they did a CV on it. So I can see the cyclic voltammetry on it. The next thing they added then was this boron carbon oxynitride. So 5 microliters of BCNO. And they tested it again. And actually, there wasn't much difference from the bare carbon to the BCNO. I would actually suggest that they got a slight improvement in the signal because they actually got a bit more peak height, and then also the peaks, peak to peak separation decreased a bit. That suggests to me that they have actually put this boron carbon oxynitride onto the surface, and in doing so, they've improved the performance of that electrode. So I would say that. Now, but what happens next is that they actually put on a recombinant receptor for SARS-CoV-2, and this is something that we would do. I should just highlight CBNO, or should I say rather BCNO. So BCNO was this boron carbon oxynitride. Sort of feels like a graphene to me with it, but some of the carbons have got boron and nitrogen. So I say that successfully goes on. But then they tried to add on this macromolecule, this recombinant protein, if I can call it that, for the receptor of SARS-CoV-2. Obviously just looked it up just to make sure I did get the definition correct and what it was, so I did understand it. And then they did a BSA, and what I should say is actually when they did the addition of this recombinant protein, this cyclic voltammetry did not change. So I almost have to stop immediately and say, look, this is the problem. The carbon electrode is working fine. You've got nice CV. Your boron carbon oxynitride modification looks like it's working because there was a slight improvement in CV. But what's happening then is you're then putting on this SARS-CoV-2 RBD. The cyclic voltammetry is not changing. It's not changing. It's telling me that actually this material is not sticking on the surface. And I've sort of looked up in the literature a little bit, and I can't really persuade myself why there's a particular affinity between this RBD and this BCNO. So I feel like, BCNO actually might be quite a good surface to make that stops things binding to it, because the evidence here is actually things are not binding to that surface. And I can't really think why there's not what we call a covalent binding. If we're modifying a surface, we will put a specific chemical bond in. I don't see in your chemistry there's any bond, you're really forming any bonds. You might get hydrophobic interactions. You might get, I was about to say pi-pi bonds, but I don't think you've got those either. So I really don't think that you have provided evidence that in fact this RBD is sticking to the surface. This is really, because interesting. Next, you put on BSA, bovine serum albumin. Yeah, we do it all the time, but it doesn't stick. It doesn't modify the electrode. I really think that there's something about this BCNO. I almost want to say it's it's resisting proteins, in fact, because even your BSA, your bovine serum albumin, it's a big known blocker, but if you put that on the surface, it will stick to the surface and distort the cyclic voltammetry. It does not. And so at the end of it, you try to add your IG here, you sort of incubate it, but really you haven't it stopped. The modification of the surface really stopped at something like step number 2 when you were trying to add in the BCNO. That did work, but step number 3 then, so everything after that then is just essentially not working. And the rest of your data actually tells me the same story. So I really feel like, no, you have a carbon electrode, that's fine, that's functioning. You put on this BCNO, that's okay, that's going on. But then after that, then this RBD is not sticking. Obviously, in our own papers, we've done similar work. So you can sort of look up this paper where we've taken the carbon electrode. We functionalized it with pyrene NHS. This NHS group here is a really active group. So the pyrene is a bit like your BCNO. This is 4 aromatic rings that really like to stack on top of carbon. So I understand that the BCNO should have good affinity for the carbon. We've got similar experience as well. But what's lacking then is when you try to stick the RBD in, you don't have any kind of, I would call it coupling chemistry. You not have any chemical bonds forming. And then the antibody, so I don't think that your RBD in your case is actually sticking to the surface. We put down the BSA, no sweat, BSA, bovine serum albumin, for example, that would stick, no probs. And we would expect the cyclic voltammetry to go from something that's quite nice, which is kind of bare carbon, to something that's quite ugly and distorted. But you don't. You just actually show nothing but improvement over time. So I really think you're just not blocking the surface with these macromolecules that you otherwise want. And I can't find literature precedents to say that this recombinant protein would really stick to this BCNO. I can't find any reference for that. Question. So I'd actually follow this paper, I suppose it would be my advice, as opposed to what you're doing at the moment doesn't look like it's actually working. So I would actually change chemistries to try and get that recombinant protein to stick to the surface. Question number 10, he's got 2 parts really. He wants to measure pH and oxygen on an Arduino. I'm really only going to mention the pH part because this guy's doing his masters. He hasn't got much time left. I would generally suggest just concentrate on pH. I have got a source here for open source potentiostats. There is a comment there about making a potentiostat from Arduino. I know it's not what you're looking for really, but just FYI. We do have a note on measuring pH with an Arduino. We don't support this, by the way. What I mean by that is we've put the literature out there years ago. The page stays active, and you can find it, and I've essentially referenced it here. But it's not something that we're sort of actively supporting. It's just a kind of community service. But the pH sensors that I recommend, you were looking for flexibility, so hopefully I have put a link to this. So these particular ones I recommend, and here's a link to it as well. Total antioxidative status. TAS is a way of measuring the antioxidative status. You can use this in sort of human health, veterinary health, food storage conditions. So TAS, total antioxidative status, is a good way of measuring the antioxidative status something. But what you'll find is that we've obviously tested it, for example, between 0 and 10 millimolar of a molecule called Trollox. Trollox is a good molecule for sort of being a surrogate for understanding the oxidative molecules, oxidizable molecules in your sample. So we've tested it with Trollox. The workflow is really the same as before, that we have this, And we have functionalized these sensors to be task sensors. We have our apps, we have our cloud, we can put the sample on there. The app can tell the meter what to do, and the data can go to the cloud, and we can put millimole equivalents of Trolux, as an output into the cloud as well. Question number 12 rather is biosensors for blood gas analyzers. So at ZP, many of our sensors come in this form factor. It's not the only form factor we do, but I do think they make a good form factor for people doing blood gas analyzers. In fact, I know that companies like Radiometer, which do make a good blood gas analyzer, use technologies like this in their blood gas analyzers. And the sensors that we have are sodium, glucose, potassium, lactate, chloride, calcium, pH. I actually think we have more, but I let people go through the list, which there's a link here to sort of find the sensors that maybe they're particularly interested in. What's nice about blood gas analyzers is a lot of what I talked about today has been sort of discrete measurements. You take a sample, you put it on the sensor, get a measurement, throw the sensor away. In blood gas analyzers, you tend to put more samples over the same sensor. And a lot of blood gas analyzers actually have wash solutions and calibration solutions on board them. So I'm taking that into account when I'm sort of talking about this, that our sensors are pretty good for people working with blood gas analyzers. And the idea that you can have like a sensor in a flow stream, you can have the sample, which would be a carousel with all the patient samples on there. You can have wash solutions that can be brought to the sensor or pumped to the sensor, and we can sort of do calibrations upon that and washes upon those. So that actually should work really well. But there's a big thing that we really have to understand what people's equipment is capable of in order to sort of do that marriage between what we have and what people's equipment is. So I think it's really a progression of micro projects, mini projects, proof of principle projects, and minimally viable products. So I think people have to engage us as engineers in order to say, right, these are the sensors we've got, that's the hardware you've got, what's going to be the strategy in order to get our sensors into your product, and engineering services can be bought using the links here. I think these are links to many of our kind of blood gas type sensors that I've put here. I did have a question 13. So measuring sodium in the lab. This came in through last night, and I realized that I've put it off the list, or I left it off the list. But measuring sodium in the lab. So one of our videos online shows one of our scientists taking a sensor, putting it in a beaker, and being able to measure sodium in the lab. In order to do this, you need the sodium sensor, so I put a link here. You often need a banana connector. So a banana collector, when I connector here is, this will go between the potentiostat and the sensor. I do have a picture of it. You also need a potentiostat, and you also need test solutions. That's a little bit faint, but I have definitely put the link underneath the video. So you have sensors and connectors. You need this anapot, which is will connect into the connector, and then the sensor itself goes in the front of the connector. And then I've also put some test solutions in there as well, just so you can get going. Otherwise, people have not a mess. They have a terrible time getting going because they're making up all sorts of strange solutions. So we do hope that kit will really help people. So I think that finishes with a kind of thank you. But if I just do a quick summary, there's also a lot of questions that have come in this week. And I just want to kind of wrap them up a little bit at the end here. Yeah, here we go. So sensors for Troponin I. Yes, we can do it. It's probably best done with a project. Platinum group metals. Yes, we have that meter. We have the sensors. We have the app. And we have the cloud. Operational life of a CGM over 125 days. Cyclic Voltammetric Stripping for PCB. It's a very similar technology to the platinum group metal technology. Technologies for Propofol. These are all similar technologies, except now we're targeting an organic molecule as opposed to metals that have also been discussed here. Custom screen printed electrodes, always a real problem because quality comes with time and effort. And so when you do customization to start with, there's not much time and there's not much budget. So unfortunately, the quality does suffer a lot. The fructose sensor operates at 0 to 20 millimole. If you need to go higher, just do a dilution. Sampling system, how to bring sample to sensors. Functionalizing of screen printed electrode. I really believe these people are not actually functionalizing. I can't see any chemistry reason why the recombinant protein is sticking to that BCNO. I actually think it's not. Measuring pH, I've given you a link to an Arduino board that will do it, and I've also recommended a flexible pH sensor for you. Total antioxidative status, really the same kit as everything else, but the sensor is functionalized to TAS. And then sensors for blood gas analyzers. Blood gas analyzers often have flow systems in them and calibration solutions or wash solutions in there, and that's really good because that allows you to then reuse that sensor and do recalibration on it. But really need to engage as an engineering team, or be paid to engage as an engineering team to really help fix the gap. And then I'm so sorry that I forgot to add in the list, because it came in so late last night, about midnight, about the sodium sensor, but I did cover it as well. So really, if you have any questions of ZP, don't hesitate to reach out to us. We do this webinar every Thursday at 8 a.m. London time. At least we do super try to do it, and you can see that we kind of put a lot of effort into making sure that we have answered people's questions. So hopefully that contact button was our contact link also makes its way underneath the video. But if you have any questions as EP, don't hesitate to reach out to us. Okay, must run to meetings, but have a good day. Thanks very much.